asking permission to record. But welcome to our February Wisdom Study Community Forum. These community forums happen every month, the last Monday of the month at 4 o'clock Pacific time. We'll make sure if you've registered for today, that means you'll get notifications for all future forums, which is great. You'll use the same login information. And if you haven't done so already, please introduce yourself in the chat. We'd love to see who's here. I see a lot of people who have been here in the past, which is amazing, and also some new participants. So thank you for being here today. So on the next slide, a couple more housekeeping items. We do want to hear from you. These are meant to be interactive. We'd love to hear your questions either in the chat or you can raise your hand. You can also speak up. We'll be looking at the chat throughout. So if there are questions we think should be answered right away, we can interrupt and make sure those questions get answered in real time. We do ask if you're not speaking to please mute yourself. And again, the meeting will be recorded and shared on our YouTube channel for those who aren't able to join. And at the end, there will be a short survey that will pop up. Please do fill that out and let us know if you have any feedback. So thank you so much for being here. So the mission of these community forums really is to move together toward equity and better outcomes in breast cancer for everyone. We started these forums over 2 years ago in an effort to really have better communication and conversation with the community, to understand what questions that you have and to learn from the community what topics matter most to you and what information might be helpful to learn more about breast health. So we have varying topics, and I think the next slide will show some of those topics that we've had in the past forums. And today we'll be talking about understanding breast cancer genetics and polygenic risk scores. This is something we've touched upon in other forums and want to take a chance to do a little bit of a deeper dive. And this will be part of a sort of a two-part series. We'll continue the discussion on polygenic risk next month at the end of March with another speaker, and we'll go through that towards the end. All of those topics can be found on our YouTube channel of all the past recordings, so we'll be sure to share that around as well. All right. So today's session, I'm pleased to introduce you to Elad Ziv, who is a primary care physician, and his research work has focused on genetics and epidemiology, specifically around cancer. So he will be talking with us today about understanding genetic risk and polygenic risk scores, both with some background on how this is used in general research in medicine, but also specifically how we're using this in the wisdom study as well. Before passing it off to Dr. Ziv, I did see a question in the very, very beginning about what the wisdom study is, and we will be going through that at the end of the presentation, but just as a quick summary, the wisdom study is a national breast cancer screening study where we're comparing a personalized screening approach to standard annual screening. And in that personalized screening approach, we take into consideration a whole host of different risk factors and do a comprehensive risk assessment to understand your personal risk of developing breast cancer, and then recommending a screening frequency that matches your risk. And so one of those components of the comprehensive risk assessment is genetic testing. So we'll go through sort of what it is, what the approach is, and where we may be looking to go in the future with learning more and being able to apply what we know about genetic risk in the future. So with that, Elad, we'll have you start presenting. I think you've got your slides that might be easier to share.
Can you hear me now?
Yes.
Okay, great. Thank you. Hi, everyone. Let me just make sure I get my slides in here. Okay. And you can see I'm in present mode.
Yep.
Great. Okay, so thanks for having me, and I'm going to talk a little bit about genetic risk. I'm going to cover some background first, and then talk about the polygenic risk score that we're using in Wisdom, because it is one of the things that we've really introduced into clinical use. So first, I just want to kind of say a few words about kind of what genetics is. The genome, as many of you know, is composed of DNA. DNA is a series of chemicals. We call them bases. And there are four types. We have short names for them, A, G, C, and T, or letters represent them. And the bases, the A, G, C, and T, really act like letters in the sense that they actually give information. The series of the letters gives information to the cell on how to make proteins. So that is sort of basically how DNA works, why it's important. And then the part that comes into effect in terms of our health, including breast cancer, is the fact that we don't all have the same sequence. We have very much most of the same sequence. Over 99.7% of our sequence is identical at all of these bases, but there are variations, positions where some of us have on one of our chromosomes an A, and others may have a G. And those spelling changes, if you will, sometimes lead to differences in how much of this or that chemical the cell makes, which sometimes then translates into differences in terms of our disease risk, and including breast cancer risk. So that's kind of the genome in a nutshell. And I have a picture here, sort of a book, and it really is like that in the sense that, well, it's not exactly a book, but there's a very long string of these bases, and there are regions that are like pages, and the smaller regions, each of them make up a gene. Okay, and each gene is one protein or one chemical that the body makes. And so a variation will typically affect only one gene. Okay. There's one other wrinkle with cancer that's important, which is that there's genetic variation that we inherit, right? So basically, for most of our DNA, we have two copies, one from our mother and one from our father. Everybody gets two copies. But there are also, and so in this case, this child you can think of has an A and a G, and they may have inherited the A from dad and the G from mom. And so every cell in their body has an A and a G. And that's just one position out of 3 billion in their genome, and each of their cells has those letters. There are also acquired genetic variants, things that we are not born with, but actually arise as we develop from childhood into adulthood, or as we age. There are new acquired genetic variants or mutations, and some of these occasionally will affect our risk of cancer also. So you can imagine that, for example, you have this one G here that becomes acquired, and then this second G then becomes acquired again. Each of these little lightning rods is a new mutation, a new change in the DNA that's acquired through life, and then that becomes cancer. Okay. This is a schematic. There's many genetic variants in the genome and so on. And sometimes actually the inherited ones can cooperate in a bad way with the acquired ones. And that oftentimes is how cancer arises, is that we have some inherited genetic risk, and then some acquired genetic mutation that then creates cancer. The other thing that I think many of you are aware of is cancer runs in families to some degree, not perfectly, and this slide will talk about that a little bit more. So, and these figures here are how genetic counselors draw pedigrees. So each circle is a woman, and the circles that are filled in have the disease. In this case, it's cancer. So these are, and the lines represent relationships. So these are sisters, and one sister has breast cancer, and the second sister is wondering about her risk. And so what is the excess risk to a woman if her sister has breast cancer? So the answer to that, it's about twofold higher than the general population. Okay. This is over many studies and seems to be relatively consistent. What about if her mother has breast cancer? It's about the same. It's about twofold higher. Okay. And what about, in this case, this is an aunt that has breast cancer. So the mother didn't have it, but the aunt has it. So the risk is 1.5-fold higher compared to the general population. So these are all risks that are higher than the general population, but just to be clear, even if we double a woman's risk, the average risk of a woman for lifetime risk of breast cancer for a woman is about 12% or something like that. So most of the women who had a sister with breast cancer will not get breast cancer. They're at higher risk for sure, but most of them will not get breast cancer. Okay. And then we also do twin studies. We try and kind of figure out, twin studies are, geneticists love them because what we have are the monozygotic twins or identical twins and the dizygotic or fraternal twins. And the secret here is that the identical twins or monozygotic twins share all of their DNA, and the non-identical twins, fraternal twins, share only half of their DNA. They're like sisters or siblings. And so we kind of can say, well, how much more risk do you get if you go from 50% sharing to 100% sharing? And it's actually about double. So it increases your risk more, but still it's not, even if you have an identical twin who got breast cancer, your risk of breast cancer is much lower than 100%. In fact, bottom line is genetic factors influence breast cancer risk, but there's much more to it than that. And I'm not going to talk too much about environment, but hopefully someone with more experience in the environment will come and talk to you at some point in the future. Okay. I think many of you have heard about BRCA1, and that's a gene that was discovered in 1993. It's associated with familial breast cancer. Women with a pathogenic variant, that's what we call the spelling changes that cause cancer. We call them pathogenic variants. In the past, they were called mutations. That, for various reasons, geneticists didn't like that terminology, so they've changed it in the last few years. So women with a pathogenic variant have a high risk, and we'll talk about how high. The pathogenic variants are transmitted 50% to each child, and that's independent of sex. But the risk of breast cancer is obviously related to the child's biological sex. So let me. And then we have BRCA2, or BRCA2 was discovered in 1995, also associated with familial risk. There are other genes, TP53, PTEN. There's intermediate risk penetrance genes, so PALB2, ATM, and CHEK2. Sort of the list has grown. These genes do not have as high an absolute risk. So BRCA1, BRCA2, we'll talk about their absolute risk. P53 and P10 are rare, but their risk is very high. as well. They're kind of like BRCA1 and BRCA2 for breast cancer, although they cause other types of breast cancer. PALB2 is a softer kind of version, as are ATM and CHEK2, but they are definitely associated with increased risk. And increasingly, when you go to the physician or to a genetic counselor and get your test, those tests are being ordered as well. And so it's important to know about them. Okay, so what happens when you get a genetic test? Usually what happens is clinicians order a gene panel. And in fact, in Wisdom, we have a 9-gene panel. So those of you who are part of Wisdom and either randomized to the risk-based arm or self-selected into the risk-based arm will get or have gotten this 9-gene panel. And then you can either get a benign variant or no variant, meaning no variant means there's no spelling changes. Basically, it's the same as the reference human genome, whoever that person was. Benign variant means, yeah, there's a spelling change, but doesn't change anything about the way that protein works. So it doesn't really matter. Then the other opposite extreme, you can get a pathogenic variant, and this is a positive result. This means this is a high risk variant. And then the other kind of annoying but important thing to know is that there are variants of uncertain significance that come back. These come back when there's a new spelling change. It's low frequency enough in the population. We haven't seen it enough yet to know that it does or doesn't cause breast cancer. The vast majority of these are eventually reclassified as benign, meaning no excess risk, but we can't know for sure. And so we have this uncertainty. So among cases, sorry, I just need to move my move the window here. So among breast cancer cases, even among breast cancer cases, most women come back with having a benign or no variant. Variants of uncertain significance is about 10%. It can be up to 30%. And that depends on the number of genes tested. The more genes we test, the more likely we are to find a new variant that no one's seen before. And then among cases, about 5 to 10% of cases have a pathogenic variant that's in a known breast cancer susceptibility gene. And then how about women without breast cancer? So more have the benign, still about 10 to 30%. This is more, I would say, probably 10 to 20% in most panels, but some of the panels that the clinical geneticists send have like 30 genes or 50 genes, and then that number becomes higher. And then in the general population, among women without breast cancer, there's still about 1 to 2% who have a pathogenic variant. Most of these are not in the BRCA1 or BRCA2. They're in these sort of intermediate penetrance genes. But we do see it, and we've seen it in Wisdom, for sure.
And Elad, just to restate this again, so the first column of numbers, so these are women who have breast cancer or individuals with breast cancer.
Right.
So of breast cancer patients, about 5 to 10% of them have a pathogenic variant.
Right. Right.
And in the general population, so people who have not had a diagnosis, it's about 1 to 2%.
Right.
Okay.
Right. Is that clear? Sort of. I think. And so the thing is, again, most women with breast cancer do not have a BRCA1 or BRCA2 or CHEK2 or ATM mutation. They or pathogenic variant. Sorry, I slipped there.
One other question. I know
Yeah.
in the early days, getting genetic testing if you had a breast cancer diagnosis wasn't as common, and it's becoming more and more common now. Do we expect that number of 5 to 10% might go up if there's more genetic testing?
No, I don't think so. I think that's a fairly stable number, and it's a pretty wide range, 5 to 10%. Part of that range has to do with which panel is used, but also part of it has to do with the age of the woman and any other risk factors. So if she has a family history or something like that, then the numbers go up. It's about, like in unselected cases, I would say it's probably about 6 or 7%. Okay, so what about the risk of breast cancer? So among women with a pathogenic BRCA1, BRCA1 variant, these are the estimated risks by age. So, and what you can see is there's very little breast cancer before age 25. But once you hit your mid-20s, the risk does begin to climb. And by age, a little after age 50, over half of women with BRCA1 have developed breast cancer, and that continues to climb throughout life. It climbs more precipitously with BRCA1 than BRCA2, but BRCA2 also climbs throughout life. The average risk is a little bit later. And so both of these genes, there are many, many different ways that they can have pathogenic variants. And each of these pathogenic variants is very, very rare in the general population. But cumulatively, less than 1% of the population has a variant, has a pathogenic variant. There's a little bit of differences across different populations, and some have slightly higher rates. Okay. This is PALB2. It's less well appreciated or less well known gene. It works actually very similarly in the cell like BRCA1 and BRCA2. And the absolute risks are a little bit lower than BRCA2. So by age 50, let's say only about 15% of women have breast cancer with PALB2, but by age 80, up to about 40% of women have had developed breast cancer with PALB2. And then I don't have the figures on here, but the other the other genes that are found commonly in the population are CHEK2 and ATM. The risks are lower with CHEK2 and ATM. So they're probably on the order of absolute risk of 20 to 30% lifetime risk with those genes. So most women with, so just going back here, most women with BRCA1 and BRCA2, by the time they hit their 70s, have developed breast cancer. Now, that's in kind of unselected populations that have not been treated or gotten any preventive therapies. Obviously, if you've gotten preventive therapy, tamoxifen, or if you've had preventive surgery, those risks will plummet very precipitously. With preventive surgery, basically, the risks are close to zero after the surgery. So these are kind of extrapolated if you don't do anything. Okay, and then so PALB2 is lower, and with CHEK2 and ATM, most of the carriers of the pathogenic variants do not get breast cancer. So the risk is higher, but it's a sort of a different thing than BRCA1 and BRCA2. Okay, which is a good introduction to the fact that there's a lot of genetics that can increase your risk, but not, it doesn't act like BRCA1 and BRCA2. It just sort of modifies your risk. And this is a slide that's meant to sort of say kind of what we know about genetic risk in the population, kind of in a general sense. And on the x-axis is how many people have a particular variant. And on the y-axis here is how much it increases your risk. And so we've already said that kind of the BRCA1 and BRCA2, there's very high risk, but the variants are very rare. The sort of PALB2 and CHEK2 and ATM are kind of in the middle here, but the variants are still pretty rare. Then there are common variants, and this is getting into polygenic risk. Common variants are things that basically we all have that increase your risk just a little bit. And these have just been appreciated, I would say, in the last 10 years or so. And they may only increase your risk by 1.05 to 1.3 fold. I mean, it's the kind of thing, it goes from 12% to 12.5 or 13%. It's the kind of thing that, who cares, really? However, the catch is that we've gathered enough information from genetic studies to know that now there are hundreds of these. And so it's really not about getting one or two, but about getting many of them, and how many. And that's really the essence of polygenic risk. And the way this is, if this slide is meant to just be a little bit educational, it's not, you certainly don't have to know anything about this slide, but just where do polygenic risk variants, where do these common variants come from? They come from very large studies, tens of thousands. Actually, there's some of the studies have now gotten to over 100,000 women with breast cancer and 100,000 controls, women without breast cancer. Investigators have genotyped, checked the genetic variants across many positions in the genome, millions of positions in the genome, and seen, are there slight differences in the frequency among cases and controls? And that's something called genome-wide association. And once we've done that, we can identify, as I said, we've identified over 200 at this point. Now, what do these, how does sort of this affect familial risk? So in terms of the proportion of familial risk that we've explained, like how much of that twofold risk in relatives have we explained, BRCA1 and BRCA2 explain part of that. It's about 20% of that. The other genes I talked about explain less, P53, P10, PALB2, CHEK2, and ATM, they explain less, but they explain, maybe a few percent. The common genetic variants, even though they're very common, their risk is very subtle. And so they don't explain a lot of familial risk. It's estimated they explain maybe another 15 or 20% of familial risk. So what this means is, even as of today, we've explained only part of familial risk, okay? And so there's a fair amount of familial risk we haven't explained yet. It has to do with the limits of our technologies in terms of doing genetic studies, and maybe some of the limits of how we're thinking about the genome, but that's not for you to worry about. But what that means is, even with all of the genetic information that we can collect, and we do collect it in wisdom, there's familial risk we haven't explained, and so we have to take that into account. Okay, now what about, how does polygenic risk work? So there's 200 genetic variants or more that affect breast cancer risk subtly. If let's say the A is the low risk and the G is the higher risk, you can either inherit 0 copies of G, 1 copy of G, or 2 copies of G, because there's 2 parents and 2 positions, 2 chromosomes. And so what that means is, in the general population, there will be people who inherit 0 copies in many of the positions that increase risk. Probably not all of them. It's just hard from a statistical standpoint to basically get 0 risk variance at 200 positions. But you can imagine that there's going to be, you can get up to 400 of the risk variants. There might be women with only 40, and they'll be at lower risk. There might be women with an intermediate, and they'll be at an intermediate risk, and then there'll be women with more than average, and they'll be at higher risk. And that's how polygenic risk works. And these are some polygenic risk score papers. And what these investigators have done is calculate or project the absolute risk of women just based on their polygenic risk alone. So this is the top, this orange line is the top 1 percentile. It's like we take, this very top 1 percentile here. Can you guys see my cursor? Okay, so we take these women at the extreme right end here, and this is their projected lifetime risk. So they're getting up to greater than 30% lifetime risk. So that's around the range of what we said. It's maybe a little higher than CHEK2 or ATM, those kind of intermediate penetration genes. Nothing like BRCA1 and BRCA2 yet. And then, there's obviously a full range. And then there are also women with low risk. So there are women who have much lower than 5% lifetime risk, which is good for them. And really what this sort of allows us to do is kind of give you a broad range. And what it also forces us to think about is that a lot of what we do in cancer screening in general is based on age. Right. So because we know age is a really strong risk factor for cancer. And so we say, OK, when you get to, and there's debate about this, when you get to 40, when you get to 45, when you get to 50, you start breast cancer screening. But the key thing here is that the polygenic risk score, we know at the top 1 percentile of risk, the risk, these investigators used a UK kind of average risk. And in the UK, the threshold for screening is about 2.5% 10-year risk. So if your risk is the average woman gets to that, I think probably in her kind of 40s or so, late 40s, early 50s, I think. And so what they're basically saying is, there are women who are reaching that in their mid-30s or even sometimes a little bit earlier. And there are women who never reach that. So we're not saying don't screen for breast cancer based on polygenic risk. But what we are doing is sort of in this range, certainly between 40 and 50, we're going to look at polygenic risk to think about whether to start screening. And actually what Wisdom is now doing is we're starting to talk to women and to invite women in their 30s. And we're also going to use polygenic risk to sort of say, well, when should you start screening? The other thing about polygenic risk is we can also predict a little bit about estrogen receptor positive breast cancer and estrogen receptor negative breast cancer. So breast cancer is not exactly one disease. There are multiple varieties of it. Most breast cancer is estrogen receptor positive. These tend to have more favorable prognosis, although not always. They tend to be treatable with drugs that target the estrogen receptor. And we can also actually quantify risk for estrogen receptor negative breast cancer. This is a breast cancer that we have fewer treatments for. It's fortunately more rare. So even in the top 1% of the polygenic risk, their lifetime risk of estrogen receptor negative breast cancer is only about 4%. But yet if we know a little bit more about a woman's risk of subtype of breast cancer, we could also potentially think about whether to screen a little bit differently, because the estrogen receptor breast cancer tends to happen a little bit younger, and it is certainly faster growing. And so it's more likely that it will happen between mammograms. And so this is something we're trying to incorporate into Wisdom. Family history is still a risk factor. So this is polygenic risk now. Sorry, the figures are not mine. The other people made them, and so I can't. They're slightly different. This is now the top 20% in the dark blue, and the bottom 20% in the light blue, and then the intermediate risk women. And what you can see is that without family history, you can stratify fairly well, but with family history, everyone goes up. Okay, and so this is kind of what I said earlier, is that family history is still a risk factor, even after all of this polygenic risk stuff. It's not a complete risk factor. So women with family history and the lowest polygenic risk are still lower risk than women without family history and the highest polygenic risk. But we have to combine them, and that's what we're doing in Wisdom. And so we're actually not just combining them with family history, but also with other risk factors that we know are important. So mammographic density and age, obviously, and history of breast biopsy are all fairly good risk factors. And we're using a model that was developed in over 1 million women. So it's got a lot of cases, and there's a fair amount of diversity in the cases and the women who were included in this cohort. And this is how our model performs. So this is the model we're actually using. It's called BCSC. And what we wanted is this number to be fairly close to 1. And this is compared to all the other models that are out there. So it's the closest to 1. And what we want is this number to be as high as possible, farthest away from 0.5. And this is also is slightly better than the other models. So this is our baseline risk model. So we kind of plug every woman into this risk model. And then what we do is add the polygenic risk on top of that, or literally maybe multiply the polygenic risk on top of that. And that forms the risk for the average risk woman, for a woman who does not have a variant, pathogenic variant in one of those genes that we talked about.
And Elad, one question in the chat. So I think this is a question about whether the tests are within the US or Scandinavia. Just to confirm, if the question is about the BCSC, the 1 million individuals in the BCSC are based from a US population.
Yes. Yes, that's the Breast Cancer Surveillance Consortium, definitely US.
Okay.
No, I don't think any of these models came from Scandinavia, but most of them. Yeah.
And then the polygenic risk score, I guess it's more the development of, or the baseline genetic data sets are international, correct?
The polygenic risk score, yes, they are international. They were actually mostly done, the lead group was in the UK. We'll talk a little bit about the different populations that are going into polygenic risk score and how we're working on that in the next few minutes. Okay, so polygenic risk score. So we put them together, and I think this is just to show that they do work together. So when we put them together, they add information. This is how the mammographic density predicts risk. And this is how the polygenic risk score predicts risk. This is by quartile or 25% groups. Okay, and then this is another paper that sort of did the same thing. So there is a bias in genome-wide association studies. This is actually taken from a paper looking at many genome-wide association studies, not just breast cancer. And the bias is European ancestry populations have been way overrepresented compared to the global population. So this is, the red is European ancestry populations by genome-wide association study over time. These colors here are basically the global population. And what you see is the European populations were overrepresented. There's this dramatic increase in how well they were, or how overrepresented they were. And this actually has to do with the UK Biobank coming online, and basically, you know, they're UK, they're European. They're not all European ancestry, but they're vast majority European ancestry. So that's an issue that I think we have to contend with in the United States. Now, just because the studies were done in Europeans doesn't mean they don't generalize, but we have to check that, and people have checked that, and I'll talk about that in a minute. And for breast cancer, but I would just say that, you know, this slide is now a little bit out of date, and things are better. In particular, some of you may have heard about All of Us, and some of you may have even participated in All of Us, in which case, thank you. The All of Us study is a US-based study, and knowing kind of what's been going on in genetics, the investigators from All of Us have really tried to overrepresent US minority populations who are underrepresented in all kinds of biomedical studies, and they've done a fairly good job. So African Americans are about 20% of All of Us, and Latinos are about 20% of All of Us. So that's not where we really need to be, like, because we need to catch up so much here. But yeah, it's getting better. And the other thing I would say is that for East Asian populations, there are large studies now in Japan and Taiwan that are have caught up, I would say. So this is a little bit better. How does the polygenic risk for breast cancer work across different ancestry groups? This is a paper from 2021, so it's a little out of date. In the green is the European ancestry. So this is how, and what the middle group is sort of the average risk group. And what we want is the lines to get higher as we go in the right and to get lower as we go to the left. That does happen. It definitely happens in the green group. It happens more in the green group. So we see attenuation in all of the populations. By attenuation, meaning it's a little closer to 1 in the highest risk group, which is not what we want. We want the highest risk group to be high risk. So that does represent a problem with polygenic risk. It doesn't mean it's not effective. In other words, the risk is higher in the high risk group, but it means it should be more effective than it is. So how are we trying to fix that? So one just technical issue is that in Wisdom now we have the option of technically including up to 20 million genetic variants in the population. And so that will just allow us to be more flexible about which genetic variants to include in our polygenic risk score models. And we are doing that on everyone going forward. And we're actually going to go back and do that, particularly on the women for whom we are concerned that they didn't get as good a polygenic risk score the first time. We got money from, do we have it yet? Or I think we selected to get money from the Department of Defense Breast Cancer Research Program to go back and get the additional information for the women from underrepresented populations, so that we can now update their polygenic risk. And the other key part is we can't really do this ourselves because we need tens of thousands of cases from different ancestry populations. And the Wisdom is big. Fortunately, most women won't get breast cancer. So we need other partners to do this. So there's a very large consortium called the Confluence Consortium. It's run by the National Cancer Institute, and we've been working closely with them. There's the LAHENA, which is a Latin American genetics consortium. It's run by Laura Feherman, and we work closely with her. And then there's an African American breast cancer consortium, which is actually made tremendous strides. It's mostly unpublished, but Fumi Olapati, who's at University of Chicago, and Dezheng Hu, who's also at University of Chicago, they are actually leading that, and they are working closely with us. And in fact, I think Dr. Olapati will be here next month.
March.
So, yeah. So I think the bottom line is this needs to improve, and it is improving. And we have the capacity within Wisdom to fix it. So, which is a good thing. I think I'll stop there. Just to summarize that there's the known breast cancer susceptibility genes BRCA1 and BRCA2. There's strong risk factors. The pathogenic variants in these genes are rare, but women with pathogenic variants are at very high risk of breast cancer. There are other genes, PALB2, CHEK2, and ATM, that are considered intermediate risk. Pathogenic variants are still rare. And women with pathogenic variants are much higher risk than the general population, but lower risk than BRCA1 and BRCA2. There's about 200 common genetic variants that are associated with very modest risk, but together they identify a small group of women who are at high risk. And really kind of what they do is sort of fan out risk across a very wide spectrum. Family history is still important because we don't know all of the genes that affect breast cancer, all the genetic variants that affect breast cancer. And so we need to count all of those things. And I'll stop there.
Thank you, Elad. As you were going through, I think one of the tables you showed was sort of how different risk factors contribute to someone's overall risk. And I think that just sort of reinstates what you just said, which is there are lots of different risk factors. They all contribute sort of differently to someone's overall risk. And so it's important to evaluate them all and look at them all together, which is what we're trying to do in the wisdom study. And there's a lot of new areas that we can explore. Again, trying to refine our polygenic risk score to be more accurate for women of non-European ancestry. And then you started to touch upon this, but another area is what other changes in the DNA could happen because of environmental factors or exposures. And that's another area with our new DoD grant that we're hoping to explore as well.
Yeah, I also think the other thing that we would eventually like to do, which I think will be helpful, is that the mammogram itself offers risk information. We use that a little bit, or we use that some. We use mammographic density as the radiologists read it, which is a strong risk factor for breast cancer, but it's not the only source of information about risk in the mammogram. And increasingly, different scientific groups have used all kinds of artificial intelligence tools to extract risk information from the mammogram. Those risk factors are really quite impressive, I think, in how well they work. And so that is something we would like to eventually incorporate. I think we need a little bit more, let's say, time to sort of just study them and integrate them in with everything else that we're doing. And knock wood, hopefully we will. We're as part of the next cycle of wisdom, we all will try and work with colleagues in different institutions who have large databases of mammograms to kind of integrate that into our models.
Yeah, that is another topic we have on the books, or hopefully soon, is a colleague at Kaiser in Northern California who will present on the role of AI and mammograms and risk prediction. So yeah, lots of very interesting topics that I think wisdom is unique, and that because we have such a huge cohort of participants and sort of the infrastructure, we can start to ask these additional questions, whether it's environmental exposures or it's looking at artificial intelligence and risk prediction. It's important that we are flexible and able to incorporate new scientific evidence into what we're doing so that the findings that we have are relevant and can really contribute to the field. One of those changes Elad just mentioned. So last year we started getting low pass whole genome sequencing data. So tens of millions of these single nucleotide polymorphisms, or SNPs, that we can use for a polygenic risk score. Before, we were just getting about 200 or 300. So again, trying to find ways where we can be flexible and really allow for the greatest knowledge to be generated. Yeah, let's see. I'm trying to think, are there more questions from the group? I think one person had a question about CHEK2 and risk by age, like age over time. You had that graph for a couple of other genes, but in general, does risk change over time as well?
Well, yes, so risk definitely increases with time for all genetic variants. CHEK2 is a little tricky because actually there isn't really one CHEK2 pathogen. For BRCA1 and BRCA2, we believe that that even though the pathogenic variants are different, in other words, the misspellings are different in the gene, once you get a misspelling, it sort of works the same way as any other misspelling. What is clear about CHEK2 is that there's subtle enough different spellings that some people with a CHEK2 pathogenic variant can have higher or lower risk than other people with a CHEK2 pathogenic variant. So sorry that that's more complicated. So I think what I said about CHEK2 is it sort of approximately doubles your risk, but it's not quite right because that's the average pathogenic variant. There is enough differences in them that some of them are actually not less than a twofold risk, and some of them are a little bit higher maybe than twofold risk. I don't know if that answers the question.
I think there's another question similar. So does check 2 risk need to be added to family history, or do you need to consider both?
Yes.
Or check 2 if you have a mutation. Does it matter if you had a family history as well?
Yes, it matters. So people have found that if you have a check 2 pathogenic variant and you have a family history, the risk is higher than if you have a check 2 variant and do not have a family history. We are counting that.
Can I ask how you calculate that? Because I have both. My sister had breast cancer, and we both have check 2 mutation.
I think what we're not. I guess what I would say is that what we're doing is affecting. So if you have check 2 and you have a pathogenic variant, a pathogenic variant in check 2, and you have family history, we're recommending a more aggressive screening program, basically with MRIs. And whereas if you're check 2 and you don't have a family history, and you don't have other risk factors, we're not recommending MRIs every 6 months. So I don't know if that answers your question. We for all check 2 carriers, we also try and get them to consider tamoxifen as well, because most of the cancers that check 2 carriers develop are estrogen receptor positive. Even more, most cancer is estrogen receptor positive, but for check 2 carriers, it's even more so. And tamoxifen reduces the risk of estrogen receptor positive breast cancer preferentially. So that is something that we ask women to consider. Basically we recommend MRIs, tamoxifen, and for that combination.
Yes, and I do both of them. Thank you.
Okay.
Great. Other questions? There was one other, I think, question about someone made a comment that their breast cancer wasn't genetic, tested twice. And I think one question that I would have is, the most times in clinical care, women or individuals are tested for the main gene, pathogenic variants, the BRCA1s, the 2s, the PALB2s. But I think one point here is that this polygenic risk is something we're looking at in wisdom and isn't offered in clinical care.
Right. Well, so most women with breast cancer come back. Only 5 to 10% have a pathogenic variant in any of the known 9 genes, and the panels vary. The one we use has 9, but there are panels with more genes, although the relative return becomes much, much lower when we add the different genes. So it's not uncommon. In fact, it's the general case that a woman who gets a panel comes back negative. On the polygenic risk perspective, every woman's got some sort of polygenic risk, right? Basically, sorry, I'm flipping quickly, but every woman, falls somewhere in the spectrum. Cases tend to be higher, but they're not all higher. So there is, right? So there are cases tend to be kind of more in the red zone, but there are cases in the green zone too. And so, and that just has to do with the fact, 2 things. One is there's a fair amount of genetics we don't know yet. Even after all the studies that we've done, we know a little about 40% of the genetics, but there's still a lot left. And not all breast cancer is going to be just influenced or a result of a genetic risk. And when we say genetic here, I mean heritable genetic. There's also the kind of somatic. Those of you who've had breast cancer, sometimes your physician will sequence the tumor itself, which is looking at the non-inherited mutations that arise in the tumor. So I don't know if that answers that question.
Yeah, I think sometimes we get the question of, I have a genetic or a pathogenic variant. It's guaranteed that I'll get breast cancer. And I think what you noted is, no, each gene has a different level of risk. It's not a sure thing that you'll get it, but it's important to know because there are prevention options that you can take into consideration. I think the other thing that we hear people say is, I have a family history. I'm definitely going to get breast cancer. And the answer to that is also not necessarily. There is an increased risk, but it's not a guarantee.
Right.
And then I think the other thing we hear is I have no genetic mutations, I'm free and clear, or I have no genetic mutations, I have no family history. So there's not one answer, there's not one risk factor, and that's why it's important to kind of consider everything as a package. And as we learn more, we can add to that package and refine someone's risk estimate.
Right.
And risk changes over time. I think that's another interesting point, is that as we learn more and our algorithms change, also as your risk factors change, it could be that you're a year older or that a new biopsy or a new family member, risk changes over time. So it's important to sort of stay engaged. And in wisdom, we do reevaluate your risk every year. And so if there are changes, we'll communicate that back so that you can change your screening regimen.
Right. That's exactly right. I think for those of us who spend our days doing genetics, we're desperately trying to sort of add, more genes to these panels or add more genetic variants to these polygenic risk scores to sort of make them as accurate as possible. And hopefully with some success, we'll kind of, it'll get better. Our ultimate goal will be that family history becomes perhaps close to irrelevant because we can read the genome so well that it sort of the family history is not as important anymore. We're nowhere near there. So we will, for wisdom and clinical practice beyond wisdom, family history should still be counted.
I think one or two more questions in the chat. There's a question, do you know if they're considering changing the screening guidelines down to 30 or 35 since there have been so many updates over the years for risks? And I think that's right where wisdom comes in.
I think so the screening guidelines are, always a moving target, and it depends on who is writing the guidelines. The American Cancer Society has until recently been 45. I don't think it's changed until recently. The U.S. Preventive Services Task Force had been until 50. They've kind of put up preliminary recommendations that move it to 40. The American College of Radiology has always been 40. So far, as far as I know, in the general population, no one's moving it beyond younger than 40. I think that I think, from a genetic perspective, it makes sense to us that, it's worthwhile knowing that, BRCA1 and BRCA2 or PALB2 variant, and even the CHEK2 variant in your 30s, because that is when that's when that risk begins to increase for those genes. And so if you're going to get it, might as well get it in your 30s. And because the polygenic risk score is going to be a lifetime thing, we figure we'll just get that also in the same time. And so we've moved wisdom to the 30s. It would not necessarily mean that mammography should start in the 30s, but some sort of genetic and familial risk assessment can begin in the 30s, probably should begin in the 30s. And then beyond that, we should think about when to start mammograms and maybe even to use the mammogram itself as a piece of information and say, well, your mammogram looks low risk and your genetics looks low risk, come back sometime, many years from now, because it's all good. Or conversely, like, ooh, there's something looks high risk here, come back soon. You don't have cancer now, but come back soon. So I mean, that's essentially what wisdom is about, is sort of making age into just one risk factor, not the whole story. And also, this is my personal pet peeve, but there's nothing magical about 40 from my biological perspective, or 45 or 50 or 35. Like nothing happens magically. Well, in your late 40s or early 50s, you have most women go through menopause, but the point is that risk is really kind of approximately linear with age, but that's only one risk factor.
Yeah. Let's see, there was one last question. I know we're over time. So one person said that her PRS score is 0.56. And so she's just questioning, or does that reduce her 5-year risk? And the answer is yes. In wisdom, we combine it and it does lower your risk.
So the way we calculate the polygenic risk is we try and standardize it to 1 in the general population. And so basically less than 1 is lower than the general population risk, and the lower the better. So and higher than 1 obviously increases your risk.
Awesome. Well, great. I know we're over time. Thanks everyone so much for joining. So this is, again, our sort of background precursor. Next month, we will have Fumi Olopade from University of Chicago speaking a little in a more detail around ancestry-specific genetic risk and risk across different populations. As Elad said, her work has really focused on women of African ancestry and working with some big consortium to try and refine our understanding of genetic risk as well. So she will be here, and we hope that you can join us. It will be on the 25th of March. Let me just double check that. At the same time, 4 to 5 p.m. Pacific time, and your registration that you've had for this webinar will continue over to the next one as well. I do believe we have a quick evaluation that is in the chat. Please take a look at that. Send us your ideas, your feedback. We'd love to hear how you liked the discussion today, and if there are any ideas for next time as well. So with that, thank you so much, Dr. Ziv, and thanks everyone for joining. Thanks for your great questions, and think of some good ones for next month as well as we continue this topic. Thanks so much.